Today, I'm going to be speaking with Dr. Eric Topol, who is founder and director of the Scripps Research Translational Institute, also a professor of molecular medicine and executive vice president at Scripps Research. It's so great to have you on. I really appreciate it.
Oh, David, great to be with you.
So let's maybe start with vaccination and kind of center our conversation there. We have currently two approved vaccines in the United States under emergency use authorization for coronavirus, the Pfizer-BioNTech vaccine, as well as the Moderna vaccine. There are other vaccines that have started to be approved in other countries, all currently, correct me if I'm wrong, mRNA platform vaccines. There has been much made of the short amount of time it has taken to get these vaccines. This is something that is merely a function of the platform. Is that right? It just takes less time to develop a vaccine under this paradigm.
Well, a few things just to clear up there.
Yes.
So, yes, the mRNA platform did work very quickly within days of having the sequence of the virus that was released from the Chinese scientist in January. Within days, there was already a template for an mRNA vaccine. But there are some other vaccines besides the two that you mentioned that have gotten approval on an emergency basis in other countries that are not mRNA. So, for example, the AstraZeneca is an adenovirus vector, not an mRNA. Basically, and a couple of the Chinese firm and the Russian Sputnik, these are different. They're using viral proteins rather than the mRNA, which is basically just giving into the muscle a message to make spike protein that's modified instead of the virus. So it's perfect because you don't have to risk any virus. It's just that one component, which is an excellent target. And by the way, David, all the vaccines do have that common thread. They use the spike protein as the primary target.
Is there anything, what is important? I'll say it more broadly. What is important for the average vaccine recipient to know in terms of the differences for them in receiving an mRNA-based vaccine versus the weakened or dead virus-based vaccine?
Right. So, basically you have two trials that came out first that were very large, 44,000 with the Pfizer-BioNTech and 30,000 plus with the Moderna. They both had 95% efficacy. They both had really good safety profile without concerns there, at least out to 2 months. And that should cover most of the concerns on safety. So that's kind of the new reference standard for the other vaccines. So the AstraZeneca at the main dose that it's been tested so far had an efficacy of 62% using a viral vector. And then others have come in at variable levels, but they haven't had as large trial experience. So what you really want as a vaccinee, a person getting a vaccine, you want to know how high is the efficacy, how well was it tested, was it in really large trials with good follow-up. These are important concerns because we're seeing some reduction in threshold for getting approval because obviously we're in a very dire situation right now.
Yeah. When we hear about numbers like 95%, as you are talking about, that would suggest that out of every 20 people that get the vaccine, one will not develop sufficient protection against an infection. Is it possible to know through an antibody test whether you are in that 5%? And if so, would it be potentially something you could do in looking at getting another vaccine? If you get the Moderna and you're in the 5%, can you go and get the Pfizer and maybe you'll be in the 19 out of 20?
Right. That's a really great question. And actually, I share your interest in having a neutralization antibody test. We don't have one that would tell you for sure that you're protected.
Yeah.
Because it isn't 100%. In the Moderna trial, which was interesting, for severe infections that would require hospitalization, it was 30 versus 0. So it looked like it was 100%, but those are very small numbers.
Yeah.
But so one thing is you want to be protected, and it's not yes or no. I mean, you want to be protected from, most importantly is severe infections, but you want to be fully protected. There's another layer of concern here that isn't settled, and that is you could get the virus in your upper airway, particularly your nose, nasal lining, mucosa, and you could pass it on without that. This is that term of mucosal immunity or sterilization immunity. And so we don't have a full readout on that yet. We have some encouraging preliminary data. So yes, you're right. If we could find out the people who didn't get the ideal response, you could try another vaccine. That's right. And we don't have any, commercial assay to get the answer to your question, David. I wish we did, because then you would, about 2 weeks or a month after you got your second dose,
Right.
you would get a checkpoint. And then if you weren't high levels of neutralization antibody, you would get another vaccine. That would be the ideal scenario, but nobody's talking about that. You're the first one that's really brought that out.
Yeah, and when you start doing the math, if you say, well, what are the odds that if you've got a room of people, I mean, I think if I understand the math correctly, if you have a room of 20 people and you vaccinate all of them, there's a 5% chance one has no immunity. And is it correct to say there's a 2.5% chance that 2 of those 20 end up with no immunity? I mean, is the math that simple?
Well, it's not quite that precise. The reason is that, these trials were really large, and we're only talking about a tiny subset,
Right.
who were the first ones to cross the line and get infections. So the numbers, as you get much larger, could be different. I mean, this is kind of giving you a ballpark. There were highly significant differences, of course,
Yeah.
but it could turn out to be 3% or 7%. We don't know.
Okay.
There's another part of this story that's interesting to point out, and that is the trials are the ideal experience. That is, they're conducted, rigorously with how the vaccines are given. But in the real world, they may have gotten thawed out early. They may not have, been cared for. The actual sterile, the glass
Vials.
Vials. Yeah, good. Yeah, thank you. That were not kept in the deep freeze or
Yeah.
whatever. So you see, it may be a little different in the real world. It usually isn't better. It's usually a little worse, right?
So in terms of, I mean, I'll be very transparent. I think the logistical rollout so far has been pretty disastrous. There were promises of 100 million doses in 2020, then promises of 20 million vaccinated. Today, as we talk, we have about 4.5 million first doses that have been given out, with question marks around how much, if any, immunity you get from the first dose. It seems it's probably some, but we don't really know, and the vaccine makers are being careful to say it's not been tested. At what level do you believe we are going to start to see, and you can answer either or both, deaths and or cases noticeably pulled down by vaccination?
Right. Well, it has been a terrible shortfall so far, as you aptly point out, David. We were expecting at least 20 million people to have had their first dose in December.
Right.
And we're not even quite at 5 million yet. So there is an issue, of course, that their vaccines have largely been delivered. That is, there's close to 20 million out there now, but they're just not getting in people's arms,
Right.
shots. Now, to get this herd immunity, which is the only way to get to herd immunity, not through natural infections, through vaccines, we need to get, at least approximately 70% of people. But the good part in the United States, if there is a good thing here, is that we let this virus go with rampant spread. And so we have at least 60 to 70 million people who have natural infection-induced immune response.
So you
Now,
believe
again,
that's important now. So that's about 3x the official cases. You believe the real infection rate is about 3 times the official 20 million or so.
That's right. It was just published today, actually, just before we got together in JAMA Network Open, that it's at least 3.2x of the number. So we are
Wow.
at 20 million. So we're at about 65 million based on all the studies. So the point here is that we could get there faster. Go back to your really great question about what if we had a neutralization antibody to the virus, to the spike protein. If we had that and we could say rapidly, you're coming into your vaccine, you're in the line in the queue, and we tested you, and a couple minutes later say, well, you're good to go. You're good right now. You don't need the vaccine. We need to defer you until we get to 70%. Now, if we could tap into that 60 plus 65 million people, we can get there a lot faster. But if we don't do that, we're looking at, third quarter at best. That means we got to get to 2 to 3 million vaccine people a day.
A day, right.
Plus getting them all a second dose. Now, the J&J vaccine, which is the Janssen's coming out, perhaps early February, even later this month. If that clicks, that's a single dose. So that will make life easier. Plus, it doesn't require freezing. So that will make the logistics easier, and it will also add to our vaccine supply. But overall, we're not going to get to population immunity that we need. The faster, the better, especially with this new strain, this new B117. We got to go fast now. This is a race against a very challenging, formidable, highly infectious variant.
One last question about the 60 million that we believe have been infected already. My understanding is there's still a question as to the duration and robustness of the immunity for recovered patients, and questions about, for example, if you had a really mild case, how does your degree of immunity compare to someone who recovered from a more severe case? Does it last 3 months, 6 months, 9? Correct me if I'm wrong. If reinfection was really common, we would have way more examples of it if the number is 60 million. That seems clear. But what do we know about the duration of natural immunity?
Yeah. Oh, you're really on this. This is great. So the reinfection story, in the world, we have like 35 documented cases by genomics that you sequence the virus in the first infection, second, they're very different. We know that it was a reinfection. There's probably considerably more than that. But if you think about the world number of infections, it's really low. That bespeaks the issue that natural immune response to an infection is quite good. Okay, so the interesting thing is the vaccine is what we've written about as superhuman. That is, it's even better. What I mean by that is the average person, now that we've looked out long enough, has at least 8 or 9 months of protection, at least.
Okay.
Maybe even years, but at least 8 or 9 months. Now, remember, you pointed out 5% of people with the vaccine don't have an ideal response, let's say. With the natural response, it's more than that. It could be 10%, could be 12. It's more, because a vaccine gets a much higher level of these neutralizing antibodies, which is the precious response that drives your protection. No less, T cells and other, response that's part of the protection. But most of it's the neutralizing antibodies. So to answer your question, I think what we can say is, if you have a neutralizing antibody, if it's still there at good levels, you're good to be deferred. I mean, you don't need a vaccine right now. We need that for other people to get us to this, because we are unfortunately going to face a vaccine shortage. Unlike Israel, which is the lead, the pace setter, they're going to have enough vaccines. They order early. They have a supply. They have enough vaccines for everybody.
Yeah, I believe they're at 14% already.
Yeah, actually, that was as of the 3rd. As of today, they're going at least 1.5% a day. So they're probably at least 16%. They've now have all the vaccines they need to get everybody vaccinated who's willing, which is most people, of course, there. Unlike what we have, which is some hesitancy, resistance, anti-vaxxers. But even with our issues, our biggest problem is getting shots in the arm, getting the second doses at high scale, like we did in 1947 with smallpox, when we had 6.5 million people in a few weeks. We've got to be doing that now every day throughout the country.
So give us a few bullet points. There's so much that could be said about the logistics that could be organized, but give us a few bullet points about things that should be done right away. One anecdotal story. My stepmom works in healthcare, but not as a direct provider. She works for a company that in sort of management, I guess you would say. And she got a call on January 1st saying, listen, we got vaccines for the first responders. We have 2 vaccines left over. There's no system for us to send these to a for nurses at a different place. We just have two vaccines and they're going to go bad. Do you want a vaccine? And she said, sure. That multiplied by 100 million, it seems inconceivably chaotic. So like, what are the logistical things that need to happen here?
Right. Well, it is crazy that we're not just trying to do everything we can to get vaccines into people. And so we waste, a lot of shots. Obviously, once they're thawed, if they don't get used, they're lost. And we just don't want any of that to happen. So we need mass events. We need an all-out effort to get a huge number of people every day throughout the country. And whether that's, getting stadiums and big facilities where, just people on an hourly basis show up and quick, get their jab, or whether it's mobile, efforts, with vans and, whatever it takes. Obviously, pharmacies could help here, because they're pretty widely distributed. So the Walgreens and CVS, they should be part of the story as well. But we need, this idea that, I have high regard for the new administration, but they're talking about 100 million vaccines in 100 days. Well, that's only 50 million people in 100 days of the new administration. That's not nearly enough.
It sounds so good, and yet the math of it is still falling very short.
Absolutely, yeah. So that's just not going to work. We have to be a whole lot more aggressive. I hope that's going to change.
I hope so. We've been speaking with Dr. Eric Topol, who is founder and director of the Scripps Research Translational Institute, also a professor of molecular medicine. Dr. Topol, such a pleasure having you, and I really appreciate your time. I know you're very busy.
Well, I'm really impressed how well you're on this, David. You've really, I didn't think you had a science background, but it sure looks like it.
I don't. My formal training is in business, if you could imagine. No, but
Well,
listen, it's the issue of our time, and for my audience's sake, I must remain informed.
Well, you're a fast learner. Great to meet you and great to have a conversation.